So what my research was is about healing, ability of these cells to actually heal tissues in our body itself. Instead of actually creating an organ, can we go back and heal the tissue that is there, already diseased tissue, to actually healthy tissue again. If I want to tell you about adult stem cells, there are two different types of stem cells that are very important. One is called mesenchymal stem cells, MSCs, and endothelial progenitor cells. Those are the two cells called EPCs, very commonly called EPCs. And these cells can be isolated from any kind of fat tissue or other tissues and can be grown, and part of these tissues cells can actually grow into a small number of different tissues that we want. Again, adult stem cell therapy is not new, as we know that if you have a bone cancer, for example, people get bone marrow transplants. So what body is doing is basically bone marrow from other person is actually putting in your own bone marrow, and you can actually treat these diseases. So it's not a new therapy that we are talking about. Not only that, our body actually has a pluripotent cells that it releases. If suppose you have a disease in your body or some kind of healing is going on, if you have a cut, our body has a pluripotent cells. They are called stem cells, not exactly like stem cells, but their ability to actually differentiate into different small number of cells that actually body releases in response to your diseases. So what we want to do is, can we use these cells that body releases? Can we ask our body to actually on demand to create those cells for us? And can we use those cells to target them to go to the site where we want them to go? So these are the two questions I had. Can we create them on demand, and can we actually target them to go to where you want them to go? So I'm going to give you two different areas that I work in my lab, and this is adult stem mobilization and isolation protocol. So basically what I've given here is there are a lot of different people are working in these areas. I'm just giving you some references here, but basically we are researching two different areas in cardiovascular diseases, and I'll talk to you about both of them. As all of us know, heart and arterial conduction is very important. We talked about it. Our heart is a pump that actually pumps blood to our whole body. But not only that, our heartbeat is, if I'm putting my heart here, heart is beating. But not only that, if I might put my finger here, my artery is also pulsating. What happens is that conduction system from the heart, it goes to your arteries, and arteries are also pulsating. And we need that elastic pulsating tissue to actually work our heart and also our arteries, to actually take the blood to where you want to go. What happens is that as you age, your arteries become stiffer. As your joints become stiffer, your arteries are starting getting stiffer. You get calcification inside the arteries. Calcification is basically deposition of bone-like mineral. It's like a hard diamond-like mineral in the arteries. And think about the artery that is very pliable when you're young, and now it is become very stiff because of this calcium there. And that happens to a lot of people with diabetes people, for example. They will get actually this disease very common for them. They will get deposition of calcium in their arteries, and there is no therapy right now. If you have a disease that is calcified arteries, basically you need replacements. There's nothing else we can do. But if you think about it, our bone is made up of hard tissue, and bone is made up of two different types of cells. Osteoblast cells make bone, which is a mineral, and osteoclast cells resorb bone. If something happens in your body, your bone is the only tissue that can be resorbed and deposited back, because our bone has these cells sitting there all the time, and they deposit bone all the time. So can we use these therapies? Can we, because arteries are now have bone cells inside and depositing calcium, can we take bone cells from osteoclasts, which are the cells that resorb calcium from the bone? Can we put them in arteries and make them resorb calcium from the arteries? That was the idea to do this. So what we decided to do is take a bone marrow cells from this was done in the animals, and took those cells from bone marrow, turned them into osteoclast type cells. Osteoclasts are the cells, as is shown here, one cell shown here, that has a capacity to resorb mineral. And these are the cells that we created in the lab from the bone marrow by giving them certain signals. And we show that these cells actually work very well. They are osteoclast-like cells. That means they have capacity to resorb mineral. And we put them on a bone-like tissue. So what we did is we created a hydroxyapatite is a mineral that is in the bone. We put those cells on top of it, and they start chewing this calcium. As you can see, there are white pits that you see inside. That is actually that mineral was taken up by the cells. So that means these cells are functional, and they can actually resorb mineral that we want them to do. So the idea was to take these cells now and put them close to the arterial mineral tissue. So what we did is we took these osteoclast cells and put them in close to the calcified arteries and see if they can actually resorb calcium from the arteries without damaging the artery. And again, we got significant decrease in calcification, about 35% decrease in calcification. Again, it's not a 100% calcification reduction, but there was a significant reduction in calcium content of the arteries. And there was a scientific, this was done in vitro, that means in the culture media plate. The same thing what we did is we actually took those cells and put them in vivo close to the arterial tissue in vivo, and we injected the cells and see if those cells actually can take out mineral in vivo. And they did exactly the same thing what we wanted. They took out 50% of the calcium that was there, was taken up by these cells. So that means these cells actually can do that. The only question is that these osteoclast cells also release enzymes that can degrade tissues, because they had to degrade collagen in the bone to actually deposit bone. So we want to make sure these cells are not degrading our arteries, because we want to keep the artery intact and just mineral has to come out. But we are working on that right now, and this is just to give you a small what we are doing. Basically, osteoclasts can be differentiated from the bone marrow progenitor cells. We can deliver those osteoclasts to the site to get rid of mineral from the arteries. That's what we are trying to work on. This is one of the project that we have in our lab. The second project is about creating small tubes of arteries. And all of us know that if you have a heart attack, basically what happens is your coronary arteries got blocked. Coronaries are the arteries that provide blood to your heart. Heart needs a lot of blood to actually pump to work properly. As your coronary arteries block, you basically what you have right now is you either get a bypass surgery, that means you rob Peter to pay Paul, basically take arteries from one place to take to other place and try to replace your heart arteries. Or you take a vascular graft from, say, synthetic graft, but they don't work very well in the small diameter. Coronary arteries are very small. They're about 2 to 3 millimeters in diameter. Anything that small diameter, if you put a synthetic graft, it will fail because of the thrombosis. Our body will create a clot inside the artery and it will clot. So what everybody is trying to do is there any way we can create these small diameter tubes, and that can be used as a transplant for this patient, and where these tubes can come from. So one of the projects that we have is basically this way. My question was, can we create a blood vessel using patient's own body? And a lot of people talked about you can actually take cells from the patient's scaffold, from the synthetic scaffold, and put them in grow them in the cell culture plate. That's one way to do it. But can we use our own body to create organs? That was my idea, to use your body as a bioreactor that will create organs for you.